For years, the fight against Alzheimer's has felt like this, just this monumental struggle. But what if we've been looking at it the wrong way? What if the real solution isn't just about managing the disease, but about going right to the source, our own DNA, and flipping a single tiny switch from high risk to highly protective? Well, a groundbreaking new study is suggesting that this might not be science fiction for much longer. Okay, so at the very heart of the story, you've got two different versions of the exact same gene. Think of it like a superhero movie. On one side, you have APOE4, the villain. It's the single biggest genetic risk factor for late-onset Alzheimer's. If you have it, your risk can shoot up by as much as 13 times. But on the other side, you have the hero, APOE2. This version is so protective, it slashes the risk by a staggering 99%. I mean, it's the ultimate genetic good guy versus bad guy scenario. And this leads us to this really bold, almost audacious question that scientists decided to ask. What if you could take that risky APOE4 gene, the one someone already has in their body, and just flip it into the protective APOE2 version? Could you actually do that? To really get how they pulled this off, we need to understand just how important this one gene, it's called apolipoprotein E, or APOE for short, really is. When it comes to our brain health, it is a true genetic double-edged sword. See, every single one of us has the APOE gene, but we don't all have the same version, and the specific type you inherit can either grant you this incredible protection against Alzheimer's, or if you get the APOE4 version, it pretty much stacks the deck against you. It contributes to amyloid plaques, inflammation, just a whole cascade of problems in the brain. So you can see the stakes are incredibly high. How in the world do you even start to test an idea as wild as this? Well, the solution these researchers came up with was, frankly, ingenious. Let me introduce you to the hero of this story, the switch mouse. What the researchers did was create this special mouse that was born with the high-risk human APOE4 gene, but, and this is the brilliant part, they built in a secret weapon. Tucked right behind that gene was a hidden genetic cassette with the protective APOE2 gene, just waiting to be activated by a simple chemical trigger. So let's set the scene. At the beginning of the experiment, these mice are just living their lives with the human APOE4 gene. Their whole biology reflects the risks that come with it. This is our before picture. And then comes the big moment. The scientists give the mice a simple drug injection. And this drug, it acts like a key. It unlocks this mechanism that literally cuts out the APOE4 gene and seamlessly swaps in the protective APOE2 gene to take its place. And this happens all over the mouse's body, including the brain. The switch is flipped. Okay, but that's the theory, right? The really critical next step was to prove it. Did the gene actually switch? To find out, they had to look deep inside the brain, all the way down to the protein level. So what did they see before they flipped the switch? Well, they analyzed the proteins in the brain, and the results were just crystal clear. Before the drug, the brain was basically flooded with that risky APOE4 protein. It made up almost 99% of the total. That was their starting point. But after they gave the drug, wow, the picture completely changed. That risky APOE4 protein, it basically vanished. And now over 90% of the APOE protein in the brain was the good guy, the protective E2 version. It was a near perfect swap. I mean, this was just a massive success. The genetic switch didn't just kind of work, it worked exactly as they designed it to. Not just in one little area, but system-wide, achieving this incredibly efficient conversion from the risky protein to the protective one. But here's the billion-dollar question, right? Proving the switch works is one thing, but does it actually do anything to help with the disease itself? To find out, they ran the exact same experiment, but this time in mice that were also designed to develop Alzheimer's-like symptoms. And the results were absolutely stunning. Flipping that one switch from E4 to E2 created this whole cascade of positive effects. The mice got better cognitively, the toxic amyloid plaques went down, brain inflammation was reduced, and less of that problematic APOE protein was getting stuck to the plaques. It was a win on basically every front. But the study turned up another really fascinating detail. As they dug deeper, they found that one specific type of cell seemed to be the MVP in this incredible turnaround: astrocytes. You can think of these as the brain's support crew, and it turns out they're the main producers of APOE in the brain. So to test this, they did another experiment.
This time, they flipped the switch only in the astrocytes, nowhere else. And the result? Well, here's a quote directly from the paper. They said that this astrocyte-specific switch was sufficient to decrease multiple AD pathologies. Just changing it in those cells was enough. That tells us that astrocytes are a huge target for future treatments. So let's just step back for a second and look at the big picture here. This isn't just some cool mouse experiment. This represents a fundamental shift in how we can even think about treating a disease like Alzheimer's. Why does this matter so much? Because for the first time, we have real, solid proof that we don't just have to treat the symptoms of Alzheimer's, like trying to clear plaques after they've already formed. We could potentially go upstream and fix the primary genetic problem that's driving the disease in the first place. This study basically lays out a potential roadmap for how we could treat this in people one day. You can imagine it, right? First, you identify people who carry the risky APOE4 gene. Then, you could use advanced gene editing tools, something like CRISPR, to go in and perform that same precise conversion, effectively swapping their genetic risk for genetic protection. Now, we have to be clear, this is still early-stage research, and it was done in mice. We're not there yet. But the door is now wide open. This research establishes a bold new frontier in the fight against Alzheimer's, the ability to combat the disease not by cleaning up the mess it makes, but by fixing the problem at its very source code. And that leaves us with a really profound thought to end on. We've just seen how rewriting a single gene can dramatically reverse disease in an animal model. As these technologies get better and safer, are we standing on the edge of a new era? An era where editing our own DNA could be the ultimate key to defeating not just Alzheimer's, but a whole host of other devastating diseases? It's a lot to think about.